you've got quiz 2 and 3 over chapters 2 and 3, which is basically going to be this week's lectures, right? Molecules of life, some basic chemistry, some Breaking Bad kind of stuff, but legal, which is kind of cool still, I think. And then cell bio. I actually think this is where we start getting really interesting material, in my opinion, okay? Upper left, that's a human cell. Remember, we are bags of compartments. That's all we are. Bags of compartments. So we can separate, compartmentalize, put certain compounds in certain areas, move one molecule to another space, either take energy to do it or create energy while we do it. Everything in cell biology is kind of hovering and building around the single cell, the nucleus. We'll talk about the cellular machinery. We'll go over kind of the central dogma, how you make proteins, DNA to RNA to proteins. Again, all of this should be review. Lower right, this is actually a fluorescently labeled image of a HeLa cell. Anybody know what a HeLa cell is? Anybody know who the HeLa cell was named after? Okay. Henrietta Lacks. Any information about Henrietta Lacks you'd like to share?
She had ovarian cancer, and doctors took a sample of the cancer, and they were able to replicate it and keep it alive. But then there's ethical health.
Okay, in the back, can you guys hear any of that in the back? Probably not. She had actually cervical cancer, but very close, okay, cervical cancer. We're in the 50s, the 1950s, and her doctor took a sample, George Otto Guy, without her permission, without her family's permission. Miss Lacks passed. Her cervical cancer lived on. He found it so fascinating that these cells would live outside of the body and have this amazing ability to replicate that he gave samples to his colleagues for free. Then he actually sold some of the cells to companies that stored them. And you can buy them today. They're called HeLa cells. And her family sued and said, hey, those belong to our grandmother. And the Supreme Court of California, in their infinite wisdom against this case, decided that a person's discarded tissue is no longer their property and can, in fact, be commercialized without your consent. And there's a wonderful book, The Immortal Life of Henrietta Lacks, that I think you should read. Okay? It's kind of an interesting ethical story, right? And many of you are going to be making some of these decisions as future practitioners. What do you do like that? So we've had this fascination for years. We talked about it. In 1663, Robert Hooke, his microscope looked different than this. But when we started being able to see the cell, we started to appreciate what the power of the cell could be. And I've been enamored with cells and cell biology for years. We do a lot of stem cell research. Not cancer cell. We've done some cancer cell research, but mostly stem cell research. And so I agree with Dr. Hooke and 200 years of observations that led to the cell theory in the early to mid 1800s, 1838. And these observations of the cell theory characterize kind of 4 components about cells. That they're the basic structural and functional unit of life, and they share generalized features. Number 1. Number 2, organisms are made up of one or more cells. Some would argue, okay, what about an amoeba? Isn't it an organism and is it single cell? There's always going to be some exceptions, okay? But generally speaking, these 4 principles apply. Cell functions are carried out by subcellular structures called organelles. We'll learn a little bit about those or remind you a little bit about those. And lastly, all cells arose from pre-existing cells. the basic tenets of cell theory. So what's in a typical cell? Whether it's the HeLa cell line or a typical fibroblast out of your skin, okay, cardiac cell. What's a typical cell made of? Well, you've got a plasma or a cell membrane. That's the outer boundary of the cell. Remember, we talked about how we're kind of bags of compartmentalization. The cell ultimately compartmentalizes things, separates the inside from the outside, or the intracellular fluid inside the cell from the extracellular fluid outside the cell. Inside the cell, we call it the cytoplasm. The fluid is called the cytosol, and the stuff inside the fluid, or the organelles, together make up the cytoplasm. So the difference between cytoplasm is you got cytosol plus organelles equals cytoplasm. There's a decent review in your textbook on the different organelles. We're not going to go over them ad nauseum, but I do want you to be familiar with them. And the way I want you to be familiar with them is maybe in this context. I want you to think about or appreciate the cell as a protein factory. It's a manufacturing facility. And the point of the cell is to make products. Most of the products that the cell is going to make are actual proteins, like what we talked about on Monday. You manufacture them. You manufacture them from a recipe. The recipe is the DNA. The DNA is found in the nucleus. The DNA is transcribed to what? RNA. Okay, specifically messenger RNA. Messenger RNA translates what? You go DNA to mRNA to proteins. Okay? Just keeping it simple. These proteins are actually packaged in an assembly line, like this endomembrane transport. And it moves this protein in its package either inside the cell somewhere, like if it's actin, for example. Actin is a protein that's found as a cytoskeletal protein. So it's made inside the cell, and the cell packages it and moves it within the cell to a destination where it needs more actin. We're going to look in the muscle physiology section at how actin is organized inside a muscle cell. But all cells have actin. Now, what about collagen? I mentioned collagen on Monday. It's packaged and it's excreted out of the cell into the extracellular tissue. And it's the framework by which your cells crawl upon, live upon, exist upon. So a question that I pose is, a typical human cell is about 10 to 15 microns. Micrometers. A frog egg, about 1 to 3 millimeters. The largest cell we've described is the giant squid. It's about 39 feet long. Or if you're measuring in diameter, we've got an ostrich egg. Long axis is about 6 inches. So why are our cells in our body so small if there are clearly examples in nature of larger cells? What's the benefit to create smaller cells inside of you and I? There has to be some biologic benefit to spend that kind of energy biologically to make that happen. Yeah. It's more efficient. You have a small volume. It's easier to control a small volume. You need to alter pH. You can do that more quickly. You need to create more actin inside that cell so that you can contract the muscle more quickly. It's easier to upregulate the expression of actin. Did that statement just make sense? Upregulate the expression of actin? Right? You tell the DNA, code more messenger RNA to make more actin protein because I need to put more actin inside the cell. If the cell's 39 feet long, it's going to take a while for actin to be manufactured for a cell that's 39 feet long. But if you have thousands of cells that make up that 39 feet, and each of them have cellular machinery doing their part, right? Many hands make light work. Make sense? Like if all of us decided to clean up this classroom, everybody picked up one piece of trash, right? And this isn't even hypothetical, right? Think about doing this. Wash your hands on the way out when you're done. But when we leave, if everybody grabbed like one little piece of trash, the staff here would come in and go, wow, these are the cleanest students on campus. If Jenna and I have to do that at the end, it may take us like 15, 20 minutes, right? Take all of you a split second. So it's more efficient to have a smaller cell. Because they're so small, we have to have these microscopes to see them. And hence, we introduced cell biology, Robert Hooke. You can either fire light energy, visible light energy, or you can fire other particles like electrons. So across campus in the Wetha building, we actually have a imaging and histology core facility that I oversee. And we have some electron microscopes in there where instead of shining visible light, we actually shoot electrons at the sample. The sample is coated with an electron reflective surface. The electrons bounce off. You collect the electrons in a detector, and it reconstructs the image. And that's how you get SEM pictures, scanning electron microscopes. Okay, I've got a video that I want to show you because, where'd it go? There it is. This video is the fluid mosaic model that we talked about. Let's see how well this will work.
Living machinery of the inner cell and the harsh conditions of the outside world stains the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins, which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients in waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
All right.
Let's say I'm a biology student working on a...
Good for you. He is a biology student. Okay. Did that video kind of help portray what I was trying to explain with the pool example and the styrofoam balls? And isn't it amazing that the delicacy of that membrane, that cell membrane, I don't think is well described in some of your prior classes. It's not very robust. It can fracture pretty easily. In fact, it does. And then that's why you actually have to make new cells, right? So you overtrain, right? You are outside for too long in the sun. You're around maybe some contaminants or harsh chemicals. You overexert yourself. There's damage and trauma to your tissues. Those cells die every day, and our body manufactures replacements. So, like, a cell is not here for the rest of your life. It typically, at some level, except in the central nervous system, You manufacture new ones. You make new ones. So the amazing thing about this plasma membrane is this fluid mosaic model and the functions that it does so well, even though it's so delicate. And these still pictures just don't do it justice. That's why I wanted you to see kind of this goofy video, I know, and it's got this stupid music, and it puts you to sleep. But the concept I want you to visualize is how fluid it is and how easily it can be disrupted. But yet, it's efficient because our cells are so small, there's so many of them, and while you're sleeping is when most of your cell metabolism happens. That's when you make new cells, while we're resting. You slow down many metabolic processes throughout your conscious life activities. It allows your body to repair itself, right? That's why when I wasn't feeling well last week, mostly what I did was sleep. And it replenishes the cells that are damaged and traumatized. So they serve as barriers, gatekeepers, allow certain things through, keep other things out, creates shape and support, as well as communication from one cell to another. These are all functions of the plasma membrane. A great possible multiple choice question, right? Which of the following are functions of the plasma membrane? A, B, C, D, right? Could be all of the above, so don't get thrown off on that. What are some things that can enter the phospholipid bilayer? Because this thing is absolutely semi-permeable. So let's back up one second. This is a scanning electron micrograph of the phospholipid bilayer right here. This is kind of this dark band. And we talked about how you've got the polar heads that are the phosphate group, and you've got the non-polar tails, which are the neutral acids, and they organize the tails in, right, because they're hydrophilic, so they don't like the water. And embedded within the membrane are these transmembrane proteins. And we talked about that swimming pool example with the styrofoam balls, where you could grab any one of them and move it on the surface. These membranes aren't stuck here, meaning they can't go right or left, but at some level they're anchored inside the cell, so they have limited flexibility. Right? They can kind of move back and forth. They're not going to really go, like I said, from the deep end of the pool to the shallow end of the pool until or once they're tethered. If you manufacture them early on, you can move them around, but once they're tethered to anchoring proteins, they're going to stay put. So what gets through? Well, some things that are small enough can cross. Some things that are small enough can cross. And they're going to go through either channels, which are openings, or if they're hydrophobic and have a carrier protein, they can actually embed into the membrane and then be dragged through. But for the most part, you've got this way to gate certain things coming in and certain things coming out. So these membrane proteins, they look really interesting. Like here's a channel. You've got the ability for an opening here. You've got the closing on one side. This is a transmembrane protein that spans the entire width of the membrane. You can see that we've got embedded proteins throughout this membrane. You've got glycolipids, glycoproteins, carbohydrate chains. You even have cholesterol that's embedded in the membrane to provide extra support for the membrane itself. All sorts of different types of membrane proteins. Most of these are sugar proteins, a sugar group and a protein group, or what we call a glycoprotein. We've got a sugar coating typically on the extracellular side of the plasma membrane. And the functions that we have of these different types of proteins are for transport, recognition, enzymatic activity, like we saw with the disaccharide breaking down into two monosaccharides. Cell-to-cell connection or linkage, receptor activity, and cytoskeletal attachment. So this is a cell adhesion molecule, CAM, from one cell to another, kind of making a handshake. Can you guys appreciate that? So that's kind of the function or the way that your immune system operates. Is you've got immune cells in your body. They have plasma membranes. They have proteins on their surface. These proteins specifically are looking for foreign objects. You have sentinel cells that are cruising through your body trying to find an invader. And when it finds it and attaches like this and has cell-to-cell linkage through a cell adhesion molecule, it'll signal that other cell that's foreign for death, for termination. And that's how you fight infection, disease, viruses. It's the whole function of your immune system. So again, very basic building block stuff in this unit, stuff that you've already had. But again, if we don't refresh it, and I can't encourage you to understand kind of the value and the importance, when you move on, like let's say you take my pathology class, we have the whole immune section, and you can't appreciate how these transmembrane proteins see foreign antigens or foreign invaders, you're going to be so lost. But once you see and understand and appreciate sort of this baseline, then you have places that you can build from. These proteins that we manufacture inside, all of these proteins are made inside the cell. There's DNA that codes for them, turns into messenger RNA, translates a protein. You're like, one of these giant proteins, Dr. Keller? Yeah. And then it signals it, putting it in a vesicle and says, go fuse with the plasma membrane and embed the protein right there in the plasma membrane. Every single one of these. If it's actin, keep it inside. If it's collagen, excrete it. So the cell is deciding what products it sends, out of the cell, keeps in the cell, and embeds in the plasma membrane. It's pretty sophisticated. If we look at these proteins, they have different categories, like fibrous proteins or globular proteins. Fibrous proteins are going to be structural, rod-like. You'll see fibers. This almost looks like rebar. I think that's what collagen looks like. It looks like these pieces of metal that you put when you actually do concrete work. Right? You can't just lay down concrete without any rebar reinforcement. Like in Phoenix, you ever see like the big overpasses being built? And they have like this twisting metal column of wire, right? And you're like, what the? That's the inner architecture of rebar that they're going to actually infuse concrete into and around, and that creates the structure. That's very, very similar. They stole that straight from the human body. That's exactly how we're made. So if it's a rigid rod-like structure like actin, collagen, elastin, we would refer to this as a fibrous or a structural type of protein. Another great possible test question, a week from Monday. You're like, what? Did you just say there's a test a week from Monday? I did. February 5th is your first exam. That's a week from Monday. Okay, so this weekend, we'll push out some of the study materials, the study guides, for you to start this weekend. How's that sound? Start thinking through, like, okay, how is he going to write a question on this slide? Or how is he going to write a question on the next slide, on globular proteins? But with fibrous proteins, give me an example. Which of the following would be considered fibrous or structural proteins? And I list collagen, actin. I list elastin, because you wrote it down. It's not on the slide, but I said it in lecture, so it's fair game. Right? So there's 3 examples of structural or fibrous proteins. Then I try to throw you off and I say hemoglobin. And you're like, nope, that's a globular protein. Right? Does that make sense? Okay. These guys are not very soluble. That means they do not go into solution in water very easily. That's a good thing. You don't want your rebar that makes up your tissues to go into solution, right? To dissolve. That would be bad. So these are not very soluble. In fact, in the laboratory, we actually sequence the gene for elastin, and we make it like in gram quantities or kilogram quantities. And it's a little difficult to get it to go into solution, but you can. And one of the ways that you get it to go into solution is you turn up the heat and you lower the pH. And the protein starts to unfold, and then you can actually get it to go into solution. Make sense? That's that whole pH and denaturing and changing the three-dimensional shape of a protein. You can't overdo it because it won't actually fold back. But if you do it gently enough, you can get it to unfold, go into solution, and then actually fold back together. Globular proteins. We saw this picture before on Monday of hemoglobin. We said it had 4 iron molecules that have a carrying capacity for 1 oxygen per iron. Oxygen loves iron. That's why you get rust, right? Get the iron out in the yard, readily available oxygen, wait a few days, and all of a sudden you've got rust. So that's why we have a rust-colored blood system, because of the iron binding the oxygen and the oxidation that takes place. So, structural versus globular proteins. All right, I want you to think about this question. I want you to do this work. Go to an SI session, go to a TA session, and talk through the order of manufacturing in a protein factory inside of a cell. I'll give you a hint, or we'll start the first one. Where does it begin? What's organelle number 1? The nucleus. Okay, that's where you're going to start. This will be great material either in office hours, TA sessions, SI sessions for you guys to study with, okay? Next week I'll kind of post the order if you don't have it. But this is a great review because organelles are fair game on exam number 1. Let's talk a little bit about cellular respiration. What's another term for cellular respiration? I said it before. I see you mumbling something. Aerobic? Aerobic respiration. Very good. Cellular respiration, otherwise known as aerobic respiration. True or false? It requires oxygen. True. Very good. Okay. Starting to come back? Starting to remember some of this stuff? All right. If you've got a printout or if you've got it on your tablet, see if you can actually put an asterisk next to where glycolysis takes place. Or think about where does glycolysis take place within a cell. What part of the cell? You know it's inside the cell somewhere because I told you that. Yep, answer right here. In the matrix, not for glycolysis. Hold that thought. In the cytoplasm. Glycolysis takes place in the cytoplasm. How about the Krebs cycle? Where does the Krebs cycle take place? Mitochondrial matrix. And the electron transport chain, this takes place in the inner membrane space. So you can kind of see on this diagram, this isn't a finger. And my kids are like, that's a gross looking finger, Dad. This is actually the mitochondria. And we've got glycolysis happening outside of the mitochondria, which is in the cytoplasm of the cell. We've got the Krebs cycle taking place within the matrix of the mitochondria. And then we've got this intermembrane space is where you've got the ETC. And again, like I said, I do want you to understand those things. Am I going to test you on all the intermediates, like how many NADHs and FADHs and ABCQRZ? No. I just want you to know, without oxygen, glycolysis. Faster. Nets, how many? 2. With oxygen, slower, more ATP. Nets, how many? 36. Some of you are like, 36? It kind of depends on how you count. So it's like, I'm not going to give you like a 34, 36, and 38 answer and be like, ha, 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 I got you, right? I'll make it pretty obvious on a multiple choice. Like we're going to be picking 2 versus 36. If I put like 21, it is drinking age, but it doesn't have anything to do with making energy, okay? Let's look at this diagram as we talk about some of the intermediates. And what I want you to be able to appreciate kind of at the top is we've got proteins, carbohydrates, as well as fats. And all of these have some substrate that allows for glycolysis to take place. That's how important it is. So you can burn fat, protein, or sugar through a glycolytic pathway. You first have to convert to an intermediate of pyruvate in order to fuel the glycolytic pathway. But if you have tissue where you're storing glycogen, which is the polymer chain form of glucose, right? We talked about that. Or you have fat storage. Fat's a very, very efficient way to store energy because there's no water, right? You take the water out of fat, and it has a tremendous number of kilocalories of energy. And that's why our bodies choose to store it as fat. And then protein. The last resort through a ketosis pathway is to burn protein, but you will do it to stay alive, okay? You'll become cannibalistic internally and go through ketotic processes to burn protein or amino acids. If you've got oxygen present, you'll form an intermediate known as acetyl-CoA, go through the citric acid cycle, and generate more ATP. This can continue on through the ETC as you start donating electrons. And the final electron acceptor is what? Remember? I think I heard it. Final electron acceptor? Water. Okay. Take out a piece of paper, scribe this out. Let's talk about some terminology differences. Glycolysis versus glycogenolysis versus gluconeogenesis versus glycogen. What are these terms? What are these terms? Anybody want to take glycolysis? Pretty easy, softball. We've been talking about it for a while. What's glycolysis? Anaerobic respiration, like it. What does it do? What does glycolysis do? It's a process to do what? To generate energy, ATP specifically. Breaks down sugar into ATP. Glycogenolysis. What's glycogenolysis? What do you think it is? What does it sound like? It's got the root word glycogen.
Does it break apart glycogen?
It breaks apart glycogen. The breakdown of glycogen to form what? Glucose. Perfect. Gluconeogenesis. Gluconeogenesis. Break the word down. Sugar, new beginning. Right? In the Latin? What do you think that is? Way in the back. Is it turning glucose into glycogen? Not necessarily. Gluconeogenesis, you're making new glucose from scratch. Okay? The liver is the primary culprit for gluconeogenesis. But that was a good guess on the formation of glycogen. What is glycogen? Back to Monday. Storage form of glucose. Is it a monomer? Glycogen? Is it a dimer? What? It's a polymer. Thank you. Okay, so I think you're starting to memorize or remember some of this stuff. Okay, We just kind of did that. All right, here's a possible test question. You perform a chemical analysis of a bone cell named as an osteocyte. We'll go through osteocytes later in the semester. You find that it contains large amounts of pyruvate. You determine that what has taken place? Gluconeogenesis, the Krebs cycle, glycogenolysis, two of these, or none of these. It's a bone cell, has a large amount of pyruvate. What do you think's happened inside that bone cell? What's pyruvate a result of? What was it? Glycolysis. Is glycolysis listed? No. Which one would it be? None of these. Very good. Okay, let's look at passive membrane transport, and then we're going to look at active membrane transport as we wrap up the rest of today. So passive transport. Passive transport is one in which energy really isn't required. Things are going to move down a concentration gradient, if you remember from basic bio. So the principles of diffusion play. You've got a concentration gradient. Remember, we have a solvent plus a solute equals a solution. So in any solution, you're going to have water and you're going to have particulates. In cells, we very rarely find a circumstance where the intracellular fluid and the extracellular fluid are in equilibrium. There's usually some difference between the two, unless the individual's dead. Okay? So, solutes are constantly moving in us. We spend so much of our energy, our ATP, moving things around in our body, staying organized, staying compartmentalized. Again, it's not one 39-foot long cell. It's millions of cells that make up your 5 to 6.5 feet, wherever you fall in that range, right? So, if you remember, diffusion is that solutes are going to move down a concentration gradient. So they're going to go from an area of high concentration to an area of low concentration. That does not take energy to do that. So if we look at passive transport, we see that no energy is needed. It uses the principles of diffusion. It goes from high to low. We've got an opportunity for sugars, ions, and amino acids to move either by passive transport, no energy, or by something we refer to as active transport, which is energy requiring. And that's setting up these gradients, where you move things from low concentration to high concentration. You set up that concentration gradient, so that takes energy. And the body uses kind of all different strategies to do either one of these. You also have, which takes energy, bulk transport. Bulk transport is a way of like shipping things across a membrane that are too big to move unless you actually have some sort of carrier. Proteins fall into this category, and membrane vesicles. So let's look at the mechanisms of passive transport. No energy requirement. Simple diffusion. For small lipid-soluble substances, and some of these examples of small lipid-soluble substances that can move across the membrane are things like oxygen, carbon dioxide, fat-soluble vitamins. It's one of the reasons it's very dangerous to have too many fat-soluble vitamins is because they can readily just move into your cells. There's really no control. Water-soluble vitamins, if you take too many water-soluble vitamins like vitamin C, you just excrete it in your urine. It doesn't build up inside of your cell. But fat-soluble vitamins like vitamin E, they actually will readily just get toxic levels inside of your cells. And that's why they caution you on fat-soluble vitamin intake. So fat-soluble vitamins, carbon dioxide, and oxygen, simple diffusion right across the cell. Osmosis. So this figure is showing osmosis. And this osmosis concept is where students, particularly on the next couple of slides, tend to get a little confused. So it's a great time to kind of stretch the mind a little bit, wake up. Okay, I know we're kind of at the last, about 20 minutes, less than 20 minutes remaining in class. It's 4:53 in the afternoon. You're tired. But this stuff, historically, students struggle with. It's not complicated, but it seems to really mess people up. Osmosis. If you've got the inability for the solutes to move down their concentration gradient, then the water will move. Let me repeat that. In osmosis, it's the movement of water. In diffusion, it's the movement of particles. In osmosis, it's the movement of water because the particles can't move. They're either stuck, the hole sizes are too small, the pores are too small, but water can squeeze through. So water will move to an area of high solute concentration. Water will move to an area of high solute concentration. So, for example, if you start up here in a beaker that has a semi-permeable membrane, and the water is indicated by the small blue spheres, the solutes are indicated by the larger orange spheres, and so if you could move the solutes, the orange spheres would go over here. But they can't fit through this membrane, so the water moves from the right side into the left side. And that is osmosis, the movement of water into an area of high solute concentration. Water follows solutes. That's kind of the lingo that we use. Why is this important? This is really important clinically because you could kill a patient if you hung an IV bag of pure water. You're like, what? Who would be stupid enough to do that? You'd be surprised at how frequently that happens. Okay? Saline. Right? A bag of saline in a hospital is what % sodium chloride? Anybody know? What's that? 0.9% sodium chloride. Not a lot, but that 0.9 is very different than pure water. So let's talk about tonicity. Let's talk about isotonic solutions, which is what our body is made up of. Salt water. We're going to talk about hypotonic solution. Undersalted. We're going to talk about hypertonic solutions. Oversalted. Oversalinity. You follow? When you say these terms, you're talking about the fluid, not the cell. So you put a cell inside of an isotonic solution, a hypotonic solution, or a hypertonic solution. Okay? This red blood cell has been put into which type of solution? Iso, hyper, hypo. Cannot determine from the information given. D might be true in your mind, okay, but it's not the right answer. I'm posing the question first to see if anybody can answer it, and then we'll explain it. What's that? Hypertonic solution, that's right. Why is it hypertonic solution? Do you want to answer that, or somebody else want? How about somebody different? The person behind you there. Yep.
Because the solution is going, the water is using osmosis to go into it, to float it, to balance the solid level.
So in this particular picture, this cell is actually shrunk. Okay, so which way is the water going? Out. Okay, so it's a little easier to see it in this format. Here is a bloated red blood cell. Here's a normal red blood cell, biconcave, right? It's like a donut that's not all the way through, right? So it's kind of like two saucers put together like this. And then the crenated red blood cell on the far right. That's a hypertonic solution on the far right, what we just talked about. Which one of these is isotonic? The middle one. This cell is in an isotonic solution. And then to finish out the explanation that you were saying, in this cell, what's happened? It's been put into what kind of solution? Hypotonic, so it's under-salinated, right? Hypo meaning under, hyper meaning too much, hypo meaning too little. And what's happening to this cell? Which way is the water going? It's going in. Why? Because the water follows the solutes, and there's a higher concentration of solutes inside the cell. So, right, if you gave a person a hypotonic solution, what are you going to do to their red blood cells? You're going to lyse them. You're going to rupture them. Okay, a different way of asking the same exact question. See if you can all do this. You got question 1, question 2, question 3. What happens to that particular cell that has a 10% glucose inside if you stick it in a 20% glucose solution, distilled water, which is basically what? 0% glucose, or 10% glucose? Don't shout it out. Take time to think. Write it down, and then we'll compare some answers. All right, any participants on question number 1? Anybody want to give some ideas on what happens to that cell if it's placed into condition of question number 1? Anybody? Yep. It'll shrink. Very good. Question number 2? It'll swell. Question number 3? In the very back. No net change. Nicely done. Make sense? Important to look at the IV bag before you hang it? Absolutely. Okay? Absolutely. Double check it. All right, let's look at some, in the very end moments here, we just got to finish things out. We're going to talk about facilitated, active, and bulk transport. Again, the concepts here are, Monday we talked about sort of these basic building blocks of life, right? What we're made of, you and I. Stuff that's all review, but maybe just in a light of, why do I care about tonicity, for example? Well, again, how many nurses in the audience, if everything goes the way you're planning? Yeah, I sure as heck want you to know what saline to give me, right? Simple concept, but do you see why it's so important? And somebody figured that out. That percentage that's on that bag is absolutely critically important, right? And what's one of the first things that you do when you have someone that's admitted into the hospital? Put an IV in, right? It's like the go-to. Because half the time just getting a bag of IV is going to make you feel better, right? But it's not surprising when you think about it. When we're 50 to 75% water, right, and your electrolytes are slightly off, you might feel miserable. All right, moving stuff across the membrane. We've got facilitated active and bulk transport. Facilitated passive diffusion. Facilitated passive diffusion. So this is a protein-assisted diffusion where you've used these membrane proteins to actually allow for movement of stuff across the membrane of the cell. Very classic examples are amino acids and glucose. Glucose, why is it so important? Because the cell primarily is going to live on glucose. That's an energy source for the cell. So, another clinical example. Your diabetic patient. Okay? More and more of you are going to be treating diabetic, unmanaged patients because it's on the rise. That's fact. In all the medical publications and scientific publications, we are not seeing a decrease in the incidence of diabetes. We're seeing an increase in the incidence of diabetes in every population. Okay? Every socioeconomic group, every ethnic group, it's happening across the globe. So you have a meal. You have an In-N-Out burger, right? That's coming. We talked about Chick-fil-A. We're going to have, equal marketing representation in this class. So In-N-Out's coming. You have a meal. You have your fries, right? They're animal style, whatever you like. Okay, you've got your chocolate shake. Maybe you're a strawberry shake person. After that meal, you have a tremendous amount of blood sugar floating through your body. If you can't figure a way to get that sugar into the cell, your cell cannot use it for metabolism to make ATP. And that is functionally the problem with diabetic patients. There's plenty of sugar in the blood. It just needs to be inside the cell. You've got to move it inside the cell through a carrier molecule. Insulin is the primary mechanism in order to do that. But there are other ways to do it, like with GLUT4. Okay? In fact, GLUT4 is a very potent sugar transporter. And exercise upregulates the insertion of GLUT4 in the membrane of muscle cells. And that's one of the reasons we tell our diabetic patients, you should exercise a little bit more. Because guess what? It's a non-insulin mechanism to get sugar into the cell and out of the bloodstream. Right? Helps your heart. Does a lot of great things. But a little bit of exercise in our unmanaged diabetic patients, that's why we say those things. Okay, so let's look at these. These channels, otherwise known as pores, you can kind of see them here. This is the extracellular fluid, the ECF. This is the intracellular fluid, the ICF. And we've got molecules that want to move from outside to in. The solute can bind into the binding site. A conformational change will spit it out on the inside process, right? It's very, very simple. It's kind of like a gate. And nerve cells operate this way. In fact, this mechanism, you can put a star next to this. This is actually how we're going to look at action potentials later in the semester. We're just going to move sodium and potassium through channels like this and allow for an electrical signal to be sent. So with active transport, we've got the movement of solute against the concentration gradient. It requires energy because energy input is needed in order to move the opposite direction of the concentration gradient. It's like rolling a ball uphill. Roll a ball downhill, you just drop it, let it go. No energy is required. To push it uphill takes energy. So these proteins are sometimes called pumps because they take energy, like a pool pump or a water pump. And these pumps, the primary transport is what we call a sodium-potassium pump. And it pumps how many sodiums out of the cell? 3. Very good. How many potassiums into the cell? 2. Okay, you remember this. This is such an important pump, the sodium-potassium ATPase pump. We're going to continue to use it, and hopefully you're not so sick of it. You'll see it not just on a couple of more slides here, but you'll see it throughout the semester. But if we look at this slide, active transport, we can put a sodium-potassium pump here on the basal side of this cell. This is the apical side. Basal side down here, apical side towards the top. Focus on the bottom first. Sodium-potassium pump. Seeing ATP, you cleave adenosine triphosphate. How many phosphate groups? 3. Every time you cleave a phosphate group, you liberate energy. So we go ATP to ADP, that's adenosine diphosphate, that's only 2, and you have a free phosphate hanging out here. You've just cleaved the phosphate, releasing energy. You do this to move a high concentration of sodium outside the cell. Now look at the top. You have high sodium outside the cell. You can bind it to a glucose. Sodium's going to come in down its concentration gradient and carry glucose with it. Does that make sense? That is secondary active transport. Secondary active transport, the glucose hitches a ride down the sodium concentration gradient, and you burned your energy down here on the basal surface with the sodium-potassium pump. Kind of cool. So the sodium-potassium pump maintains a resting potential where you've got higher potassium inside the cell So higher potassium inside the cell, higher K plus inside the cell, back to our periodic table. Higher sodium outside the cell, Na plus. Both of them are a single positively charged ion. So in cold weather, like here in Flagstaff in the wintertime, thyroid hormone stimulates cells to produce more sodium-potassium pumps. And as these pumps run, how many of you have ever put your hand on a pump that's running? Is it what, cold or warm? It's warm. This actually helps to establish our basal metabolic rate and generates heat energy during the wintertime for us. And your thyroid gland actually upregulates your sodium-potassium pump insertions in your cells during cold weather. 30 to 50% of all ATP in the body runs these pumps day and night. Pretty important. All right. Last slide of content, bulk transport. Sometimes macromolecules are too large to fit down some of these channels or these transporters. So we'll use macromolecular transport through what we call exocytosis and endocytosis. Exo, like EX, like exiting. Exocytosis is spitting it out of the cell. Endocytosis is actually bringing it into the cell. So you can see in a picture here, this is an example of how cholesterol is brought in to a cell if it's in the bloodstream. So you've got, on the far left, you've got the receptors that bind the molecule. The membrane itself invaginates or forms a pit. It pinches off and internalizes that molecule, known as endocytosis. If they're small molecules, we have a term for it. We call it pinocytosis, which actually translates to cell drinking. In phagocytosis, like macrophages that are part of your immune system, they do this when they're gobbling up bacteria from an infection. Phago is the root for eating. So pinocytosis is cell drinking, phagocytosis is cell eating. That's how you bring stuff in through macromolecular transport. Questions? Okay, great. Don't forget, I'll send out an announcement. Quiz 2 and 3 are due on Friday by 11:59 p.m. Make sure you're here next week for some participation stuff.